Good morning. This is Michael Dong on video episode number 22. EHS, we stand for Environmental Health Safety Practice, and also HPLC maintenance, with a focus on what can the lab analysts do in these two arenas. Here is the agenda. Three different sections. First one is Environmental Health and Safety Practice, or EHS. We talk about practice and we talk about solvent waste disposal. Second one is mobile phase and sample preparation, which is a pretty neglected area. We talk about mobile phase selection and preparation and general principle and practices in sample preparation, focusing on small molecule drug substance and drug products. Finally, we talk about HPLC maintenance procedure, focusing on the simple ones that the actual lab analysts can do. The ideas in the video came from the book chapters 4 and 7 of this book. On the safe handling of solvent in samples and waste, the biggest EHS concerns are solvent, particularly organic solvents. So most organic solvents are delivered is 1 gallon bottles. Some laboratory actually use the cyclotainer, which is like a beer cat using nitrogen pressure placed adjacent to the hood for getting the organic solvent, which is primarily for reverse phase acetylnitrile or methanol. For waste solvent, many laboratories use these type of cans uh, called just right cans, solvent can, which is 5 gallons in capacity, built from high density polyethylene with some safety evaporative control. According to a EHS regulation of best study, you need some evaporative control cartridges to prevent inhalation risk, meaning carbon cartridges for evaporative control. You need to kind of seal these wastes, and the solvent line come in from these Tygon tubings into the waste solvent. And you also need a secondary containment system. There are many newer type of HPLC laboratory that does not require these type of safe solvent, which is unsightly. A lot of new lab can use these type of waste system built from stainless steel tubing, which is directing the waste right into a huge central waste tank in the basement or on the first floor. So these mitigate the use of many unsightly waste solvents. Some general safety rules. One is expected to wear lab coats, safety glasses with size shields, use gloves for toxic and corrosive chemicals. You must use fume hoods for volatile and toxic solvent that goes for all the organic solvent. Well, for weighing, you should use a balance enclosure because most of the pharmaceutical is considered a safe bridge category 3, means they're toxic to human, and you cannot weigh it out in the open. If you weigh buffer, you can weigh it out in an open weighing balance. So these have laminar flow. You should not do it in a hood because the flow is too strong. Anytime you have safe bridge category 4 or 5, these are considered highly toxic or high potency compound, and you have to do all the weighing inside a glove box or the powder handling. Once the solid is contained in a vial enclosure or in a solvent, then you don't have to handle it in a glove box. Comments on mobile phase preparation, which is usually not highly discussed or highly published, focusing on solvent filtration and also degassing. A note for premix mobile phase for isocratic analysis. The practice is to measure volume of each solvent separately in a measuring cylinder and combine them. You should not do it in a volumetric flask and then QS to volume. Pump blending is generally preferred and encouraged. It actually does a better job than the manual preparation. The conventional wisdom is to filter all aqueous buffer, particularly ion pairing mobile phases, through a 0.45 micron membrane filter configured from nylon. Nylon is actually preferred because it works for both aqueous and organic solvent. Also, others are Teflon and cellulose acetate. So one would use these type of vacuum filtration apparatus using a rough pump with a solvent trap and a detachable filter for the membrane. It's very important to make sure that there's no cracks for safety handling under vacuum. However, for many laboratory, we actually do not filter any solvents. We mitigate, we do not do this step. The idea behind it is to use high purity buffer, such as ammonium formate at 99.999% buffer. There's no need to filter HPLC grade solvent because it's pre-filter, and there's no need to filter purified water, which are also pre-filter in the millicube type of system. So many lab was able to successfully forego with this practice. I know it might sound heretical and probably not endorsed by many of the service engineers, which have different purposes, but it really works well for many laboratory for many years. Degassing is really important for accurate blending, isocratic and gradient conditions. Most pumps have built-in vacuum degasser or a separate vacuum degasser. Typically, there's 4 solvent channels, and make sure that all 4 solvent channels are filled so that the vacuum pump will not be sucking air through one of these emptied semi-permeable membrane filters. For those of you who want to need more credential or idea on foregoing with filtration, this publication in 2014, 7 common faux pas in HPLC, would discuss some of the evidence of how that works. Mobile phase A separation by convention, A is the aqueous in reverse phase liquid chromatography. So one is supposed to use buffer or acidify or basicify the mobile phase A under reverse phase. This is actually mandatory for anything that's acidic or basic analyte, which actually 95% of the small molecule drug substance are either basic or acidic. The percentage is skewed towards basic. 85% of the small molecule drugs are actually contain a weak base, meaning having a tertiary, secondary, or primary amines. The acidify mobile phase A actually helps to control retention, selectivity, and also give you better peak shape. So you cannot just use water. Some very common recipe is 0.05% TF8, trifluoroacetic acid, or formic acid, or 10 millimolar ammonium formate. All these are mass spec compatible. One of the highly used phosphate buffer actually is highly used and probably not really called for, typically is a lot of people's belief that buffer are really needed only for critical separation. It's also my belief that buffer overuse in pharmaceutical analysis. In most cases, purely just acidifying the mobile phase is the way to go. I would venture to say that it's a statement that 0.1% phosphoric acid is probably underutilized and should be probably use more because it's great for purity assay, for raw material or for reagent uh, with UV detection actually below 200 nanometers. So it's great for universal application at a low wavelength. Obviously, this asset is not a buffer, right? So if you use a mass spec compatible buffer, then it is not possible to go down below 210 or 220. One should keep in mind the buffer capacity. In general, a buffer concentration should be kept on the low side. 5 to 10 or 15 millimolar are in generally optimum for most uses. Very rarely you need to go to 15 millimolar. The most first choice for pharmaceutical analysis for most reverse application is the mobile phase A of pH 2 to 4, meaning that 0.1% TFA or 0.1% formic acid. So all these are explained in greater detail in this white paper, which is probably one of the most updated white paper on the best practice and the modern trend in selection and reverse phase mobile phases, published in 2015 with authors myself and also Barry Boyer of AMT. Here are some guesstimates for shelf life. I mean, it's very difficult to know exactly, what is the exact shelf life of these mobile phase. So one can kind of, uh, using common knowledge, to guess that for buffer is probably about a week. If you refrigerate it, you can keep a little longer lifetime. For neutral buffer, it's definitely a little bit on the low side. And always, keep it away from sunshine to prevent accelerated algae growth. For simple acidified mobile phase such as 0.1% TFA in water, shelf life of 3 months is to be expected. For all organic solvent, at least 6 months. The exception is THF. Once you open up a bottle of THF or tetrahydrofuran, then you have to throw it out because of the danger of explosion from the peroxide. Some applications of ion exchange require piston seal to wash the salt off from the back of the piston under high salt mobile phase condition. In those cases, make sure that the peristaltic pump to feed the piston wash solution is prime, making sure that it's prime. So we mentioned a couple of times that buffer should not be stay in the system when it's not running, and should rinse it out with water before switching to organic mobile phases. A very strong note of caution that phosphate buffer, particularly high concentrations as 50 millimolar phosphate buffer, is not soluble in 100% acetonitrile. You should use no more than 85% acetonitrile in water if you want to mix it directly. Here is important information on sample diluent and sample preparation, which is really under discussed, and a lot of laboratory analysts are perhaps a bit undertrained. Very few publication on these type of procedures. So for sample, typically we use 2 milliliter vial. For any light sensitive compound, you should use amber vial. The receptor should not be a screw cap. It should not be creme, right? It should be a very thin membrane. And different vendor might have different requirements. So you're supposed to fill 1 milliliter, maybe perhaps up to 1.5 mil of sample, uh, using a disposable pipette right into these 2 mil HPLC vials. If you have very small amount of samples, such as 200 microliter or 300 microliter, you cannot fill an entire vial. So you're supposed to use, for water HPLC or UHPLC, use these conical bottom flask to put a small sample in there. For Agilent machines, you are supposed to use these type of insert, which will fit about 200 microliter or zero point 0.2 mils. The best sample diluent for the final sample in the vial is the mobile phase A, but without the acid or base additive. The default, if you don't know what it is, in cases of drug discovery type of sample, the default is 50% acetonitrile and water without acid or base. Try to stay away from methanol, which can esterify any alcohol in the sample. Now, one have to be careful about the strength of the diluent. Try to avoid very strong diluent such as DMSO and inject directly into HPLC to prevent peak anomalies, or use 100% acetonitrile, just too strong. Try to keep the diluent strength to be low. For sample preparation, for simple drug product, orally available products, which are typically tablets or capsules, one use a dilute and shoot for drug substance without any filtration, right? You're not supposed to filter any drug sample. However, for a drug product, you do need a filtration step. So typically, we do a grind to reduce the particle size. I would discuss that a little bit in more detail. And extract them. You must extract them quantitatively and filter them for any oral drug products. So you go through a filtration step using these disposable membrane filters. For some products such as controlled release drug product, which might have a polymer, a two-step extraction, perhaps the first step using some organic solvent might be required. For more complex sample, such as most complex samples such as serums or plasma type of sample, you do need some cleanup. For suppository sample, which is waxy, you also need to do more cleanup. So some of the cleanup is solid phase extraction or liquid extraction for physiological fluid or suppository samples. So for a simpler drug substance and drug sample, there is a white paper just published this year, 2024, in LCGC International. So this is a really good reference. Coming right from this white paper just published is a fine discussion, a more detailed discussion on sample preparation for drug substance. So we mentioned that it's really basically a wait and shoot type of situation and try to solubarize the API or the drug substances completely in a volumetric flask and then transfer it into an HPLC vial. So there are a well-trained QC chemist, do this quite well. And it actually, some training is required to be able to weigh small amount of references and also very small amount of drug substances, because you don't really have a lot to spare, particularly in early development. So here are the procedure. I just kind of read it over very quickly. Weighing is you transfer 25 to 30 milligram. Very rarely you have sufficient to let you weigh up to 50 milligram. It is possible but in general, you weigh around a 30 milligram drug substance sample. So there's two camps of thought. Many prefer, including myself, using these folded weighing paper, these type of paper, and you do a trifold to make sure that it's kind of shaped like a little shuffle. You weigh 30 milligram of the substance onto the paper. You can tear it, and then you have to transfer it completely, quantitatively into a volumetric flask. Some prefer to rinse it out with a extractant. Other would do a tear or weighing afterward. Yeah, so each laboratory analyst might have their own preferences, or they may be SOP. Others like to use these type of weighing boats and do it by difference do a quantitative transfer. And at the end, after extracting it, the extraction are typically done by shaking for a certain amount of time, being determined by method development with the diluent, or sonification, ultrasonicating to solidify it completely. And then you would, after the complete extraction, you QS it to volume, and then you would take a small aliquot, typically 1.5 mil of the extract directly into an HPLC vial. Typically, these type of disposable pipette is sufficient. Sample preparation for orally available drug product, namely tablet or capsules, a little bit more complex. Still mostly uh, extract and shoot directly. However, most tablet or capsule might require a particle size reduction, mostly for a tablet, meaning you crush or you do a milling process of anywhere from 1 tablet to 10 tablet. 10 tablet is probably the norm. In other cases where capsule are very soluble, you can drop them directly into the volumetric flask and do extensive sonification or shaking. So you crush them into white powder. You can quantitatively transfer all the powder, or you can weigh in every tablet weight. You can weigh, for instance, for 10 tablet, you weigh 10 tablet every tablet weight into the class A volumetric flask, and then you make sure that everything is extracted quantitatively before you do a filtration using a 25 millimeter disposable syringe filter. Typically, you would waste the first half a mil to waste, and then you express the filtrate directly into the HPLC vials. The next segment is HPLC maintenance and troubleshooting. Troubleshooting would be discussed in the next video. Um, Here we are focusing on a common HPLC maintenance procedure, focusing on those simpler procedure that the lab analyst can do. Some slogan or some guidance. The key for success for HPLC in operation is to keep your system maintained and calibrate the system periodically, typically during the annual preventive maintenance procedure. Most laboratory would have the manufacturer service engineer come in every year to do a preventive maintenance annually. It's nice to have a system to check system performance with a reference sample with documented parameter. We give you one example later on. The thing that you need to watch out for is you always try to verify things which can be easily confirmed, such as pressure. Pressure and your baseline, your detector baseline, is really important criteria before you actually do any sample injections. Check for leaks before testing samples. For older HPLC, leaks are much more common. For modern HPLC, if you calibrate and maintain system religiously, generally a leak is not a common problem. For older system, you should learn how to replace check valve, particularly some manufacturer. And also, if you have an older auto sampler using an external sampling syringe, learn how to replace that and learn how to make sure you get rid of the air bubble, which might be trapped. Definitely learn how to replace a UV source, which is pretty straightforward. Always use HPLC grade solvent, clean up and filter the sample, and make sure that your sample is weak, if possible. A weaker diluent would prevent any anomalies from distortion from a injection of a stronger diluent. Here is a schematic diagram of a very common system to kind of give you some idea on the complexity in some of the component. Here is a of a Waters Alliance pump. Now, Waters Alliance has been the dominant pharmaceutical lab workhorse for over 3 decades. So a lot of the older system is the Waters Alliance shown here. And here's the pump unit, which is can be taken out in the drawer. You just pull it out. And here are some parts, right? So these are courtesy of Waters Corporation. So here are the two pump head. Most pumps are dual piston in series. So each pump would have two pistons in the pump. This actually turned out to be a quaternary pump, right? So there's two pump heads in here. Here's the pump head, and here are the check valve. You can change the check valves are highly accessible. Here's the inline filter. Here is the seal pump wash, which is a peristaltic pump that allow you to wash behind the piston if you're using ion exchange chromatography with high salt. Here is the purge valve, which you can open it manually for dry pumping. If the pump is dry, the pump would not be able to pump liquid, so you have to draw out manually 10 milliliter of solvent to make sure the pump is wet. And also for the Alliance, you need to do wet prime quite a bit before you do your sample injection. Here is the pressure transducer. Here are some common HPLC maintenance procedure. Once again, we focus on those that the analyst can do. For the pump, learn how to replace the piston seal, particularly if you have the older system, which piston seal tend to be less reliable. Replacing the solvent sinker or the 10 micron filter is very easy. These are actually the one that you drop into your solvent reservoir. Also, inline filter are very easy to replace, and also check valve, particularly for Waters Alliance system. Learn how to replace a UV source from a UV detector. Auto sampler, you need to replace the rotor seal of the injector or the high pressure seal. But in general, for some of the newer auto sampler, it is a service engineer type of task. Also, sometimes the needle might be bent and need to be replaced. Once again, some of the modern UHPLC systems turn out to be much more difficult and generally done by the surface engineer. The exception is the sampling syringe, which is external to it. So here is a Teflon tipped sampling syringe, which is very common in older auto sampler, XYZ type auto sampler. So here are some of the common parts in a little more detail, replacing the Waters Alliance check valve, most likely in the mobile phase A pump, with a new check valve cartridge pointed in the upright direction. Here is the sinker. It's a 10 micron stainless steel filter disc, which can be easily replaced. And here is a inline filter. I'm going to go through the procedure to replace a deuterium lamp of a UV detector. And the system in this case study is the Waters 2487 PDA, which is very common. And here is the replacement lamp, which costs in excess of $1,000, right? So it should be replaced annually. So the first step to replace it if it's aging, or if you get very high noise, is to turn off the power switch. And you also need to pull out the plug uh, from the end, make sure that it's not plugged in, just as a safety precaution. You open up the front panel, and obviously before you take out the old source, deuterium source, make sure you have a new source available. And there's a electrical a plug, which is a quick disconnect to disconnect it from the power source. And then you would unscrew the two thumb screw using a special tool or your screwdriver, and you pull them out flush, meaning vertically without, leaning towards one side. Just take it out, and then you replace it with your new source. One should wear a lint-free cotton glove in the operation, and should always try to avoid touching the windows using your bare hands. Here is the flow cell cartridge. You can examine them, but it's very rarely that it would become dirty, right? In general, that is generally not a problem. So after you replace the source and connect everything and screw everything back, uh, you plug it back and also turn the power on. And remember to use your CDS, the conductive data system, to let the system know that you have replaced the lamp, because many CDS track the number of hours that the lamp has been on. Here's a schematic of once again, a very popular UHPLC, which is the Esland 1290 binary pump. Showing you the schematic. This is the pump head, and on top of the pumps is the degasser, a 4-channel degasser. Now, the binary pump allow you to mix two solvent at a time, right? Actually, you can have 4 solvent, A1, A2, B1, B2, but you can only mix 2 at a time. But all 4 solvent lines should be filled with solvents. This pump is a dual piston in series, so it actually, since each of the pump would have 2 piston, right? A low pressure piston and then a high pressure piston in there, and they're actually being driven with with a digital motor in here. So both, and show you the actual inlet check valve and some of the outlet check valve, right? So this is the schematic of the 1290. The 1290 also using an automated purge valve, so you don't have manually automated. And actually, you have sufficient power so that you do not need to dry prime it. Actually, you can pump it right in the dry state. The mixing is done by a high-tech mixer called a jet weaver, which have 2 sides. For a high throughput screening type of sample, you can use the 35 microliter side. But for most other application, for most pharmaceutical lab, you should use the 100 microliter, the larger jet weaver mixer. This table is a checklist of maintenance procedures being done on Agilent HPLC module, uh, listing what is typically being done on the column compartment pumps, auto sample, detector, so on, right? Things that they either replace or either they check on that. So this is included as a reference. Here is my summary and conclusion. We discuss environmental health and safety concerns and practices. The key is to minimize inhalation risk from solvent and from chemicals, and also follow EHS guidelines on disposing solvent and chemical waste. We discuss in some detail mobile phase and sample preparation, and show you some of the best practice on mobile phase and sample preparation using regulated testing of drug substance and drug products as an example. Finally, we talk about HPLC maintenance procedure that the laboratory analysts can do, particularly for a more older system, such as the water's alliance system. Uh, you know, one should learn how to replace check valve, replacing the external sampling syringe, pump filter, UV sources. I think UV source we talk about extensively how that can be done, and we cite case study from Waters Alliances and also Agilent 1290 system as two case studies. Once again, the two chapter will spell out some of these procedure in some more detail with references. So here are some of the key references, including the four white papers. Uh, Actually, three white papers on the three key modules, and some of the recommendation for UHPLC instrument maintenance. On that note, we completed episode number 20 on EHS practices and HPLC maintenance, focusing on what the lab analyst can do. Please subscribe to the channel for future videos, and we thank LCGCSA channel collaborator. We see you next time. Bye bye.